to our need. So we must have engineered all these things. So nothing is totally 100% natural about these cloning vectors, but cosmids are far modified than this phage. So what they are having, they are simply the plasmids. So as we say, they are called mids at the end, so they are simply plasmids. So they are much resemble like plasmids. They are having an origin of replication, which is the first criteria, one selectable marker, which is second criteria, and also multiple cloning site, which is the third criteria. But except for this, they are having a unique modification onto these plasmids. What is the modification? We simply add a cos site onto this plasmid. That's why it is termed as cosmids, right? So cos site is the only requirement for packaging into this phage particles, right? Because if a DNA stretch sequence is having a cos site, it can be packaged inside bacteriophage head. So by attaching the cos site onto these plasmids, we are ensuring that these plasmids can be packaged into the bacterial or packaged into the bacteriophage, right? And those, we can utilize those phage to infect the E. coli or any host bacterial cell onto which we are trying to transfer our gene, right? So gene transfer become easier, because utilizing normal plasmid, the gene transfer is pretty difficult, because we need to produce and provide the competent cells, right? To avoid this particular thing, we can modify these plasmids, because plasmids are pretty efficient, they are very efficient, and cosmids are efficient in transferring the gene. So if we combine these two things, we can make a very good vector. Now cosmids are really good vectors because their efficiency of transfer is very good, and also their efficiency of cloning is very good, and they also carry small segments, and their copy numbers are also very good. So you are combining the advantageous part of plasmids as well as the advantageous part of phage vectors together in cosmids, right? Now, but these cosmids are somewhat unstable, so these are the bad things about cosmids. But most of the part, you can clone large inserts about 45 kb, and also the features we have seen. The presence of cos sites permits it to in vitro packaging into of cosmids into the bacteriophage, and we can infect this bacteriophage onto this particular bacterial cells, right? So we get this cosmid structure. So this is another cosmid structure which is having multiple cloning site and all these things. But the disadvantage of that is that it is somewhat unstable and difficult to maintain. So we cannot store it for a long time because it is unstable due to any kind of phage and plasmid blend. This modification sometimes become unstable and difficult to maintain. This is the only disadvantage, but it is having very more advantages.